Hello, I'm Barry Robson of Engine Inc., Cleveland, Ohio, and I want to tell you about our internet knowledge and mining tools in relationship to artificial intelligence, in relationship to the rapid response to the COVID-19 epidemic. By way of introduction, here's the course of the epidemic in the very early stages. Relatively low and confined to China in the middle of January, but we were able to respond very quickly, 3, 4, 5 days, not only to the virus genome appearing in GenBank with our research, but also in writing the papers, and other papers quickly followed. So let me discuss methods which relate to rather unusual things, the hyperbolic Dirac net and the language Q-UEL associated with that. This idea arose for this language in particular, the idea that could there be a single language and mathematical system for unification of medical knowledge. It's pretty well agreed that interoperability is very poor in the healthcare industry as opposed to, say, in the finance and international travel sectors. Now, I do confess there are many ideas behind our system, and all of them have an important role, but the central one is Paul Dirac, who pointed out that his methods and those of theoretical physics generally should be applicable to many aspects of human thought, especially when expressed with numbers, and in our case those numbers are probabilities, the probabilities of truth of statements of knowledge about the world. Some terms and concepts: the bioengine is the industrialized big data version, probabilistic semantics is the discipline, the hyperbolic Dirac net follows from Dirac's work in our interpretation, and it's kind of a Bayes net on steroids. It's bidirectional, has cyclic paths. In other words, it's a general graph. It's not confined to a directed acyclic graph, which by definition is a Bayes net, really, and also has relationships other than just purely conditional ones. There's a quantum universal exchange language based on Dirac's work which supports that. Extracts the particular forms of knowledge extracted from the internet or text, and knowledge representation store, or KRS, is the collection of all these knowledge elements for the future to be used in inference and prediction. And gathering and using knowledge uses both an up-down and uses an up and a down approach. Top-down and bottom-up, as they'll be called in the AI fields. Build a repository of knowledge by data mining in various ways. Use that to make predictions, automated inference. Test the power of that and the sensitivity and specificity of that, and so forth. One can look at the original brackets, like describing a relationship for an electron between momentum and position. Compare a classic conditional probability, say from evidence-based medicine, which you're looking at blood pressure and glucose relationships, one conditional upon the other. But now imagine that's a Dirac bracket. What would that mean? Well, for a start, it means it looks in both directions. It's more than one probability here. So here's an example I've used of a hyperbolic Dirac net, actually serving here as a model for progression of the COVID infection through individual patients. Particularly important is the bra operator ket, where the operator is now related to a verb. It maps very beautifully to language. It's difficult to understand unless you say that, because quantum mechanics is complex. It's greatly simplified by replacing the square root of -1 by the square root of +1 h, which Dirac rediscovered and is used along with the square root of -1 in his formulations. But what's nice for us is it relates to natural language, and the maths therefore will hold. This is the system as it is now. Suffice to say that Dirac Smash and Dirac Miner are two forms of structured data mining used on medical records, insurance claims and such things, whereas Extractor to the right, working through Marple, is gathering knowledge from unstructured data, in other words, natural language and the text about medicine and so forth. Here's an example of a Q-UEL tag, which was actually generated by structured data mining of medical records. It looks complex, but that's because on the internet it's got to carry web management detail, it's got to carry the probabilities up front, it carries information about provenance, where it all came from, and things of that nature. We will concentrate on the yellow area to the right here using MARPL, but it's probably worth noticing that most of the studies used our system to go out and interact with web pages for standard bioinformatics tools to put the data in and to get the data back. MARPL was originally developed for setting exams, and actually as a robot medical student it was used to test the medical knowledge in the knowledge representation store, but it can use for other purposes here, such as a simple symptoms checker for COVID-19. Here's an example of an extract tag. We call them tags because they're really a kind of extended form of XML, in which it will include the links, not just from the text on various web pages, but from any scientific references at the bottom in the case of Wikipedia. Let battle commence. Here's the question form again, just like in a medical licensing exam for med students. But the question now constrains the searching of the internet. The candidate answers are like the queries in this case. And this and other factors will make sure that it stays on track and doesn't bring back nonsense that would just distract us and fill up the knowledge representation store unduly. Here's one at the extracted at the start of the study. We only had details about the SARS epidemic then, but we noted the close relationship with the what we call the SARS-CoV-2 virus today, and applied many of those principles. Focus on rapid response, but also on highly conserved sites in our approach. Firstly, because they've got to be important to the virus if they're conserved across many strains and even across many viruses. And secondly, they're less likely to mutate. You won't find a vaccine or a therapeutic useless in, a few weeks. Here's the system. We do the bioinformatics like anybody else does on the right. The only difference is that many times we use our system to actually interact with the web pages for us. But you can certainly use the classical approaches on the right, the tools of bioinformatics, to reproduce our work. Despite the emphasis that we have on conserved regions, I've got to say that popular targets are actually turning out to be quite rather variable by our standards. They're not so highly conserved across the strains of the coronavirus. So we've certainly studied these, but we look at the more conserved ones, including this sequence, an amino acid one-letter code associated with the S2 proteolytic cleavage site, which finally activates the virus for entry, and it's highly conserved across the coronaviruses and even beyond. Predictions using our hyperbolic direct net, remember a kind of Bayes net on steroid, is important because parts of the structure are missing in three-dimensional experimental determinations. They look like they're loops cut off here, so we can predict those, but we can use the predictions for other things too. Notably, predicting coil or loop is great for predicting epitopes, even if it actually isn't in that conformation as one would normally consider it, but also for spotting three-dimensional folds, which was also important to us. We'll talk about the macro domain briefly later. Synthetic peptide vaccines you know about. They're cleaner, they're much faster responses. They may be a little bit more expensive, but they're getting cheaper. You know the general idea: predict the epitope, a loop or something at the surface of a protein, the surface of the virus, touch it to a carrier protein to make it immunogenic, add some other factors and it becomes a vaccine or the basis of a diagnostic. Retroinverso peptidomimetics are when you take a section of the sequence, write it backwards and synthesize it with D amino acids, so it mimics the three-dimensional structure of the original virus site, but is now non-biodegradable. It's as if you switch the NH and CO groups of the backbone, but one can tidy this issue a little later, and sometimes the side chains are more important anyway. The ability to synthesize peptides, even large structures like proteins, which I've been involved in, greatly improves the this kind of approach. And there are various approaches like this one where we can actually program the required structure in. Look at a site on the surface of the virus, for example, or from a protein inside, indeed. Synthesize that in D amino acids, attach it to a carrier protein, raise antibody gated it, sequence the heads of the antibody, synthesize a head in D amino acid, and you now have a non-biodegradable substance which will interact with the original target. Much more guaranteed than the retro-inverso approach. For design, we use an ensemble pharmacophore approach as well. We believe that the virus has to conserve its information. It can use some of its active sites, binding sites, for more than one function, getting into the cell, doing things inside the host cell. So an ensemble pharmacophore, the idea is not totally novel, but it really blends several. And some existing proteins can actually be useful for those. Indeed, those involving steroids, human enzymes could inhibit the cytokine storm, which is an overreaction by the body, and we've always been aware of that issue. In this particular case of this kind of pharmacophore, there were 3 groups of molecule which were potential inhibitors. Here is actually the enzyme in question, 11-beta-hydroxysteroid dehydrogenase type 1. It will actually accept many inhibitors, including the steroid analog shown here. Here's some steroid analogs. The carbonoxolone is actually, at the top left, is actually the known inhibitor experimentally. There's even a X-ray structure for that in the protein site. Here's some in silico strong binders that we designed in earlier studies. They're ready to go. Emodin was, and some others were actually found by Chinese researchers and others to inhibit SARS virus entry. So that was very rewarding to hear. Here are some other successful studies from drugs proposals from clinical trials and non-invasive studies, which also suggest that these studies are on the right track. Here's some weaker binders. ACE inhibitors could be useful, but let's be careful. They're known to upregulate ACE2 as the receptor on which the virus enters. We're also able to adapt these predictive methods to predict sialic acid binding sites. You know, all respiratory viruses and gut viruses probably have to have these, really. The viruses don't transport magically from outside the body, teleporting into the ACE2 receptor. A lot of coronaviruses are known to have sialic acid binding parts, and they're essential to influenza and many other viruses. So we were interested, and they can be blocked, these sites, so we're interested in those. There's the prediction sites in the SARS-2 SARS-CoV-2 spike protein. You'll see above the red line is predicted sites. Here's some comparisons between uridinase on the left and the similar arrangement of the tryptophan rings on the right here. So that's looking feasible as the most probable site. Here it is as the blue lobes to the right of this structure. The most probable one is cited there. There are other highly conserved sites. In fact, macrodomains in the polyprotein are probably the strongest. It's conserved not only across the coronaviruses, but across viruses and into animals and plants. Archaebacteria and so on, archaeons contain them. So they're very highly conserved, really. You can spot them across many species. Here's the relevant protein for the macrodomain inside the coronavirus. It's involved with poly-ADP recognition and the response of the body, raising the immune response as well as the innate immune response. Here is some proposed compounds already known, which we found bind, as well as analogues of the enzyme, which is an ADP ribose 1 double prime phosphatase, monophosphatase was the original name. Here are many publications about the system available on request. Here the details are also available on request for coronavirus research, and please send any questions to this email.